Hi, my name is Zach, and I grow human brains. Well, actually, I use living human brain cells to grow small models of the human brain. We call these brain organoids. So, why do we do this besides that it's super cool? Well, let's say that you have a neurological disorder like Alzheimer's or epilepsy. What we'd like to be able to do is take the brain out of your skull, chunk it up into manageable bits, and monitor those bits for months or even years at a time. Obviously, this is a tough sell. So what we do instead is we make an organoid model of your brain and study that. The technology to do this has been around for about 9 years, so it's still under development. But generally, the way it goes is this. We start out with stem cells, which, as you know, are the cells that can become any other type of cell in the human body. Since we can derive these from blood or tissue samples, they'll have your DNA in them. We take small numbers of these, grow them in the lab, and as they grow, we give them chemical cues that say to them, become brain cells. And over about 2 to 3 months, they actually start to develop a cell diversity and structure that looks a lot like a human brain. As you can see from my slide, though, there are some problems with this, and that's what I'm working on with my thesis. The problem is that first, they don't grow very large. They get about 3 millimeters in diameter. And second, they don't really look like a fully mature human brain. My hypothesis is that this is because they don't have blood vessels. See, when we tell the stem cells become brain cells, we implicitly tell them don't become anything else. And part of the anything else is blood vessels. Now, this is a problem because your brain has an absolute ton of blood vessels. By some estimates, every single one of the 86 billion neurons in your brain has its own little dedicated microvessel that gives it all the oxygen and nutrients it needs. And without these, I think that these organoids just don't develop as well. So the first thing that I did was develop a method to grow blood vessels in the lab. I get these from the fat of rats as little fragments. And when I grow these fragments, they develop a network that looks a lot like the vessel network in the human brain. Then I spent about a year totally failing to get these into our brain organoids. But about 6 months ago, I hit on what looks like the right combination of environment and timing. And the results are really promising. The organoids with these blood vessels in them, they grow faster, they grow larger, and they structurally look more mature. So right now I'm spending most of my time looking at the cells and seeing if the cells themselves become more like mature cells in the human brain, which is what we're looking for. Along with other folks who are working on other aspects of this problem, this really positions us to develop the second generation of these brain organoids. It's going to open up a whole new range of new research opportunities in medical and scientific fields. I'm really excited about this work. I hope that I've made you a little excited about it too. So thank you for listening and thank you for your time.